Department of Microbiology with Martin Schuster.
All right, so before I get into what my project was focused on, I'm going to talk a little bit about the bacteria I studied. It's called Pseudomonas aeruginosa, or PA for short. It belongs to the group Proteobacteria. It's rod-shaped, as you can see in the picture. It's gram-negative, meaning that on a gram stain it will appear pink, and it is motile. It's a facultative anaerobe, meaning that although it can survive in environments with oxygen, it can also survive in environments with little to no oxygen. It's an opportunistic pathogen, meaning that when given the opportunity, it will infect a potential host. For example, if you have a severe burn wound or cystic fibrosis or are in other ways immunocompromised, it will infect you given the opportunity. And it's commonly found growing pretty much anywhere from soil to water and even places that are considered more sterile, like hospitals. And a little bit more about Pseudomonas aeruginosa. It produces pyocyanin and pyoverdine metabolites, which combine to form a sort of blue-green pigment when they're plated. And they're resistant to many antibiotics, which make them extremely difficult to kill when they establish an infection. And one of the reasons for this also that they're extremely difficult to kill is that they express valence factors in response to cell density, which is known as quorum-sensing, which can have the ability to infect the host quickly and release toxins that will kill the host. They can grow as a biofilm to protect themselves, which is basically sort of layers of Pseudomonas aeruginosa surrounded by slime, meant to protect the bacteria in the center, sort of, and its complete genome has been sequenced. And why is Pseudomonas aeruginosa not some other type of bacteria? Well, Pseudomonas aeruginosa is the main cause of death in cystic fibrosis patients. And once into adulthood, about 80% of all cystic fibrosis patients are infected with Pseudomonas aeruginosa in their lungs. Once again, it's an opportunistic pathogen, and it's a model organism for the study of quorum-sensing a little bit. And a little bit about cystic fibrosis. It's a recessive genetic disease, and it's caused by a mutation in important proteins that regulate the production of mucus and sweat. And most people have at least one working copy of this protein, but people with cystic fibrosis, both of the copies will be mutated. And because cystic fibrosis is incurable, it often results in lung transplantation or death for the patient. And Pseudomonas aeruginosa is related to cystic fibrosis in that it causes fatal infections, which are very difficult to get rid of once they've been established for a long period of time. And now about quorum-sensing, this is basically how bacteria communicate in response to cell density to express virulence genes. So you'll see here that signal synthase is producing autoinducers at a fairly low rate in response to the low cell density. But once cell density increases, more autoinducers will be produced by surrounding bacteria, and they'll be able to attach to the bacterium receptor, which causes quorum-sensing to activate, which lets the bacteria express their virulence factors. And this can occur between different types of bacteria, not just Pseudomonas aeruginosa. And LASI and LASAR are the autoinducer synthase and receptor of Pseudomonas aeruginosa respectively. And how quorum-sensing and Pseudomonas aeruginosa are related is that a quorum-sensing proficient population of bacteria is required to infect the lungs of patients. So Pseudomonas aeruginosa mutants with quorum-sensing will not be able to infect patients, and LASAR mutants will rise over time in the lungs of cystic fibrosis patients. These are basically Pseudomonas aeruginosa without the ability to quorum-sense. And later on in the established infection, the Pseudomonas aeruginosa will convert to a mucoid phenotype, which basically overproduces slime, and this will eventually be fatal for the patient. Very bad prognosis. And my project is basically to discover if there's a relationship between quorum-sensing deficiency in Pseudomonas aeruginosa and their conversion to mucoid. And my hypothesis is that there is a relation because of some of the following discoveries. LASAR mutants are more vulnerable to hydrogen peroxide, which is released by the immune system in response to bacteria establishing an infection. And LASR mutant presence in the lungs basically a lot of the time precedes mucoid conversion of Pseudomonas aeruginosa, and hydrogen peroxide stress causes Pseudomonas aeruginosa biofilms to convert to mucoid. And this is important because Quorum-Sensing helps Pseudomonas aeruginosa produce its virulence factors, which help it establish the infection and release toxins to kill the patient. These like attachments, releasing exoenzymes and toxins. And these virulence factors can be turned off by Quorum-Sensing drugs in development, mostly in early cystic fibrosis patients, because once they establish an infection, as I said, they're very difficult to affect. But if you limit Quorum-Sensing in the Pseudomonas aeruginosa, they can begin to behave like LASR mutants, and Quorum-Sensing will turn off, which may, if my hypothesis is correct, make them convert to mucoid, which will kill the patient even faster. And also, it's very important to have a general knowledge of these types of things in relation to Pseudomonas aeruginosa, so we can more fully understand how it works in cystic fibrosis patients. And some of my previous studies have revealed that when Pseudomonas aeruginosa is under oxidative stress from the immune system, mucoid conversion occurs, and if you place the Pseudomonas aeruginosa under enough stress, it will encourage mucoid conversion in Pseudomonas aeruginosa, but it won't be enough to kill them. And during a minimal inhibitory concentration experiment that I did in some of the first weeks of my internship, we discovered that 2 millimolar hydrogen peroxide would be enough to kill all the bacteria, but obviously we didn't want that to happen because we wanted mucoid conversion, so we used 1 millimolar hydrogen peroxide instead. And my cell culture procedure is basically this. I take some media, in this case I used LB, and in 4 test tubes, each labeled with their specific strain, I inoculated them, and 2 of the strains were labeled for use of hydrogen peroxide, and I also put 1 millimolar hydrogen peroxide into those once a day, and inoculated those and put them in the incubator shaker overnight. And the next day, on the second day of my experiment, and the 8 days following, I would inoculate them and put hydrogen peroxide in them twice a day, and leave those overnight. And each day, I would take the OD600 of the previous day's bacteria and measure it, so I would know how much bacteria would be needed to inoculate the media. And my plating procedure was basically how I found out whether mucoid conversion was occurring or not. I would do a dilution series for all 4 of the strains, the LASR mutants, wild type, and both with H2O2. I would take each 6th dilution and plate it 3 times for a total of 12 plates. And I would leave them in the incubator, and the next morning I would look at them to see if mucoid conversion had occurred. And my results were that although the colonies varied in shape and size, sometimes dramatically, they never seem to convert to mucoidy. Mucoidy has more of a white and slimy phenotype, and sometimes the slime, when they're in close proximity to one another, colonies will sort of bind together almost, but we never saw that in bacteria colonies I studied, and they were either yellow-green or an off-white, and they weren't slimy at all. One of the ways you can tell if they're slimy is you can take the lid off of the petri dish and sort of move it around under the light, but nothing really happened. And some interesting things I found were that small white colonies were growing on a few of the plates that I observed, and occasionally in large amounts, on about 2 of the plates, there were like 100 colonies that were this phenotype. And there assumed to either be a contaminant, which is more likely, and or there was Pseudomonas aeruginosa. And so I ran a polymerase chain reaction to find out whether or not they were Pseudomonas aeruginosa or the Pseudomonas. And basically it amplifies the DNA of Pseudomonas aeruginosa that is specific to that strain. It's supposed to reach a certain amount of base pairs on the gel. And I used primers to match the sequencing Pseudomonas aeruginosa, genus-specific and species-specific. And I had positive and negative controls. The negative controls were just water to make sure nothing got contaminated. The positive controls were just Pseudomonas aeruginosa. And here are the results of my PCR gel, brain PCR gel. The negative controls came out negative, of course, so nothing happened there. Nothing was contaminated, which is good. And the positive controls are there. Pseudomonas aeruginosa, everything worked out fine. They're all at the number of base pairs they're supposed to be. I think the top is genus-specific or species-specific, I'm not entirely sure. And here's the ladder to show the number of base pairs and where they're supposed to be at. And the unknown bacteria didn't show up, which means they were not Pseudomonas aeruginosa. And so in conclusion, there wasn't enough evidence to conclude that quorum-sensing and mucoid conversion are related. One of the reasons that this might have occurred was because I didn't challenge the Pseudomonas aeruginosa enough with hydrogen peroxide. We could have probably used a bit higher concentration than 1 millimolar. It wasn't very much. I think it was just a guess. And Pseudomonas aeruginosa also produces an enzyme that breaks down, called catalase, that breaks down hydrogen peroxide. And perhaps we could have added hydrogen peroxide 3 times a day instead of twice, so catalase wouldn't be able to break down hydrogen peroxide as quickly. And biofilm flow cells are the types of cells that generally convert to mucoid more often as opposed to planktonic growth, which is what I was studying, so perhaps I should use biofilm flow cells instead. And I'd like to acknowledge Dr. Martin Schuster and Brett Milby for being my mentors over the course of this internship, giving me things to do in the lab, helping me if I had any questions. I'd also like to thank the rest of the Schuster lab and the Department of Microbiology for answering any questions that I might have had, and the National Science Foundation for its funding. That was my question.
More hydrogen peroxide, dilution, what would you do differently?
I think the only thing that would be different is add more hydrogen peroxide.
Yeah. Well, you said initially you tried 2 millimolars, now it's too much to kill the bacteria, right? So 1 millimolar is like too little. It almost seems like that's such a narrow room in terms of how much you want to add there. Does it seem like, I don't know, I guess my question is, why such a narrow range? Do you think it's nothing accumulation, or do you think it's just because that was like, I don't know, I guess I'm not quite sure.
Yeah, honestly, I'm not sure. I guess it was just too much for bacteria and too little at the 1 millimolar concentration. I'm not entirely sure why there was such a big difference. Yeah, the last argument. So the question is why I didn't focus on, sorry, what was the stream? Okay, because the last side and last are the main quorum-sensing strains, and not so much RHL.
Any more questions? Let me ask you a question. What's the funnest thing you did in the lab?
What's the
Large lab fun.
I like autoclaving stuff.
You like killing things, huh? You like the smell, or just like you're getting straight?
I don't know, it was just something to do when I had downtime, I guess. And, I don't know, doing the dilutions was fun, but kind of tedious at the same time. Inoculating bacteria is always interesting. Studying the plates and counting them wasn't fun at all. And yeah.
All right. One second, all right.